Welcome everyone. I'd like to begin by acknowledging the traditional owners of the land on which we meet today. I would also like to pay my respects to elders past, present and emerging. Well, good afternoon and welcome to the May FightMND Scientific Seminar. My name is Sarah Bennett and I'm the Research Coordinator at FightMND. We hold these seminars monthly on the fourth Tuesday of the month, and last month in April, we heard from Dr. Alan Yu, Head of the Neuroinflammation Laboratory at the Florey. Dr. Yu spoke about a novel inflammatory pathway that could be of critical importance to driving MND progression. If you missed out on this presentation, it is now available on our website, so you can catch up at a convenient time. Following the talk today, we are leaving plenty of time for discussion and questions. So if you would like to ask anything, please don't hesitate to use your reaction button to raise your hand, and one of our team will enable your microphone and camera and let you know to ask your question. Alternatively, you could also type your question into the chat where our team will be monitoring and read out questions on your behalf. Please note that the chat function is only enabled if you are using the app-based version of Teams and unfortunately doesn't work in the web-based version. Please do ensure you introduce yourself and where you're from before asking your questions so we can all get to know each other. So now to our presentation for today. Professor Julie Atkin is the co-director and head of the Cellular Biology and Therapeutics Lab at the MND Research Centre at Macquarie University. Professor Atkin completed her PhD at the University of Sheffield in the UK before relocating to Australia to complete her postdoctoral studies. Professor Atkin has over 20 years of experience in researching the cellular and molecular neurodegeneration mechanisms involved in the pathogenesis and progression of MND, as well as using this knowledge to identify new therapeutic approaches. Additionally, she has 5 years of experience in the biotechnology industry across both Australia and the UK, enabling an advanced understanding of the therapeutic translation process. She joins us today to discuss some of her lab's latest work in developing new therapeutic approaches for MND. Thanks, Julie. Over to you.
Thanks, Sarah, and thanks for the invitation to speak today. So I initially gave this a fairly broad talk and was going to talk about our therapeutic studies, a new protein that we're working on, and some new disease mechanisms. And I prepared the first part of the talk and realized that was 30 minutes. So the time was up. And so what I wanted to do really was focus on some work where we've identified a novel isoform of FUS, because we've got some really interesting new data that I wanted to share with you. So I'll just focus on that today. But what I want to do is just say, thanks to everyone involved in the project. So we're obviously at Macquarie in the MND Research Centre. This is the centre here, based at Macquarie University. And this is a picture of my team. So thanks to everyone involved in this work, and particularly Marta Vidal. So the work I'll talk about today was really set up by, and she did most of the work, Marta, who was a former PhD student. And so I'm talking about our novel isoform of FUS, and it's quite interesting because unlike most other proteins, most other pathological proteins in ALS, this isoform is actually found extracellularly. So ALS, of course, is a complex and multifaceted condition. And I wanted to show you this model, this image that was from a recent review. And it illustrates nicely the sort of complex interplay of ALS. So, of course, we have ALS and healthy motor neurons that are impacted by this series of risk factors. So, of course, we all know that, the genetic mutations are a major cause. Genetic modifiers affects the phenotypic manifestation of the disease, such as the onset, site of symptom onset, progression rate. Environmental and lifestyle factors, cigarette smoking, hyperlipidemia, excessive exercise, military service, but the outer ring denotes aging, and this is still the major risk factor for ALS, and this is an area that we're interested in, and we published this review earlier this year. And what's interesting is that the molecular hallmarks of aging, of which there are 12 that are now well established, they overlap significantly with the neurodegenerative mechanisms in ALS. And we've sort of added another one of these, which is RNA dysregulation, and that's relevant to what I'm going to be talking about today. So FOS, of course, is one of the major pathological proteins linked to ALS. Mutations or variations cause about 4% of familial cases and about 1% of sporadic cases. And the canonical form of FOS is a DNA RNA binding protein with important functions in both RNA metabolism and DNA repair. So the canonical form has this prion-like domain at its N-terminus. It has RRM, several RGG domains, a zinc finger domain, and a nuclear localization sequence at its C-terminus. Obviously, FOS is normally a nuclear cytoplasmic protein that is mislocalized to the cytoplasm in ALS. And most of the, I think it's about 60% of the ALS-associated variants are present at the C-terminus. And I think FOS has perhaps been overlooked a little bit compared to TDP. I mean, we all know that TDP and FOS share strong similarities pathologically and functionally. And there's been some debate in the literature about whether pathological forms of FOS are present in sporadic disease. There was a study, 14 years ago now, from Deng and colleagues who suggested that FOS immunoactive inclusions were present in sporadic ALS, but then this was disputed in additional studies. However, I think two more recent studies have really confirmed that FOS is present and mislocalized in sporadic disease. So this is really placing FOS onto the pathophysiology of almost all cases. Some of the controversy was arisen due to the specific antigen decloaking method that was needed to see the FOS inclusions. But in this study by Kanaka et al., they showed that using other methods such as Sudan black B, these inclusions were present. So they're also quite separate from the independent of the FOS inclusions. So perhaps FOS is more important in ALS than what we generally recognise. So we identified a novel isoform of FOS. This was from a bioinformatics study, which is produced by an unusual alternative splicing outcome, which is thought to be induced by cellular stress. So we're about to send this off for publication, this study, and all the details of the alternative splicing mechanism will be in the publication. I haven't got time to go through all of this now. But basically, the novel isoform has a unique N-terminus of about 61 amino acids. So it basically lacks the whole of this prion-like domain and most of the RGG1 domain. However, it retains the C-terminus of canonical FOS, which is where most of the ALS-associated mutations are present. And what was interesting about this isoform, and that really struck our attention, was that it was predicted to be extracellular, which is quite unusual for the main pathological proteins in ALS. However, there was initially no experimental validation that this isoform existed. So that was our first task, to examine whether we could detect EC-FOS. So firstly, at the transcript level, FOS is contains 16 exons, FOS gene, and it has in the novel EC-FOS isoform, there's a predicted start codon at the start of exon 6. It then skips exon 7, and there is it joins the remaining sequence at exon 8. So there's this unique region between exon 6 and exon 8 that's not present in canonical FOS. So we designed PCR primers for this unique sequence, and we were able to detect the presence of this isoform. So we've called it EC-FUS for extracellular FUS. We detected this both in cell lines, in human primary neurons, and also from spinal cords of both ALS patients and control. So there's 5 healthy control patients and 7 ALS patients here. So this validates the presence of the EC-FUS transcript. What about the protein? So as it was predicted to be extracellular, we took both cell lysate condition medium. Of course, while it's predicted to be extracellular, it needs to be synthesized intracellularly. So we looked in both the lysates, labeled L, and the medium, labeled M. And what we found was that we could detect the presence of both canonical FUS, which is this strong band here in the lysates, but also the presence of EC-FUS, but in the medium as predicted. So this was in 3 different cell lines as shown here. We then went on, this was in collaboration with Albert Lee at Macquarie, and we looked to see whether we could detect EC-FUS in the medium using mass spec. And Albert detected 4 peptides, 3 of which were in the unique region, this first 61 amino acids, and also 1 peptide in the nuclear localization sequence. So this really validated that EC-FUS existed at the protein level. So we then looked to see what the properties of EC-FUS were compared to canonical FUS. And we found that this unique N-terminus of EC-FUS is quite different to canonical FUS in terms of its amino acid composition. So if we look at canonical FUS, the N-terminus contains mostly polar residues. However, EC-FUS contains mostly hydrophobic residues, so it's very rich in alanine and valine. So when we look at the grand average of hydropathy, we can see that this is where positive value represents a hydrophobic protein and negative value is more hydrophilic. EC-FUS was much more hydrophobic than canonical FUS. We then went on to do a Kite-Doolittle hydropathy plot. So this is where higher scores represent the most hydrophobic proteins. So a positive value up to 4.6 represents hydrophobicity, whereas a negative value represents more hydrophilic. And if we look at the N-termini of canonical FUS and EC-FUS, you can see that EC-FUS has this very strong hydrophobic region as we predicted. So this would imply that it's highly aggregation prone. So we also looked at this using various bioinformatic tools. This was in collaboration with Oscar from the University of Barcelona. And what we found was that EC-FUS was indeed predicted to be highly aggregation prone. So we used 3 different algorithms, AgriScan, CAMSOL, and AmeliaPred. So these use different parameters to assess aggregation. And what we found was that EC-FUS was extremely aggregation prone. These 3 regions where the aggregation was very strong, particularly between residues 21 and 48, but 2 other regions as well. This was entirely absent from canonical FUS, and there was small regions that were aggregation prone within the common C-terminus. We also did AlphaFold modelling, and this predicted that EC-FUS was intrinsically disordered in aqueous solution, so it would be significantly exposed to solvent and thus prone to aggregate. And if we look at the values for AgriScan, you can see that where again a positive value represents aggregation prone nature of the protein, The beta amyloid, for example, gives a value of 6.4, but EC-FOS gave a value of 23.6. So this predicts that it's highly aggregation prone. So then we tested this experimentally, and we looked in both cell lines and human primary neurons using confocal microscopy. And this is just expressing the wild type form of EC-FOS and also expressing the equivalent construct of canonical FOS. And when we compared the number of inclusions formed by each of these two proteins, you can see that EC-FOS produced 3 to 4-fold more aggregates, both in primary neurons, this is human primary neurons, and in HEX cells. So this again confirms that EC-FOS, even the wild type, is incredibly aggregation prone. We then went on and made some ALS equivalent variants. So we chose 2 common FOS mutations, R521G and P525L, which become R316G and P320L in EC-FOS. And these are both present in the nuclear localization sequence at the C-terminus. And what we found was that these ALS-associated variants further enhance this aggregation propensity. So there was a significant increase in the number of aggregates compared to the wild type. So even though wild type is very aggregation prone, these variants are even more aggregation prone. We then looked at the types of inclusions or the size of these inclusions, the area, and we found that whereas the wild type was making some smaller types of inclusions, 2 to 10 micrometer squared, basically we found that with the mutants, they were forming much more of these larger aggregates. We also looked at toxicity by assessing activated caspase 3, and we found that while wild type appeared to induce some toxicity compared to controls, these 2 mutants further enhance the toxicity. So these mutants are more toxic and more aggregation prone. So the bioinformatics suggested that EC-FOS was inherently disordered, so we wondered if it was also unfolded. And here we use this TPE dye, which is produced by Yu-Ying Hong from La Trobe. And basically, with this dye, it fluoresces when free cysteines are present, which are normally buried within the core of a globular protein. But when there's a free cysteine thiol that is exposed to solvent, we see the fluorescence. And what we found was that EC-FOS, both wild type and the 2 ALS-associated mutations were inherently unfolded. We then went on to make a specific antibody for EC-FOS. Distinct from canonical FOS, and we did this by generating a polyclonal antibody where the immunogen was an epitope in the N-terminal region, this unique N-terminus of EC-FOS. And we expressed HA-tagged canonical FOS and EC-FOS in cells, and this second blot here represents a HA blot, and you can see that both canonical FOS, the higher molecular weight protein, is present, and EC-FOS, but when we use our specific antibody, we detect only EC-FOS. It's not detecting the 72 kilodalton canonical FOS. We're currently generating a monoclonal antibody as well with GenScript, so hopefully we'll have another antibody available soon. We've also looked in primary neurons to look at the expression of EC-FOS, and you can see it's present throughout this whole cell, but also particularly this strong staining within the neurites. And this is interesting because FOS is known to play a role at the synapse, and that at least some of the studies that have detected FOS at the synapse have actually used a C-terminal antibody that would recognize both the endogenous canonical FOS and also this isoform. We then, given that we now have a specific antibody to detect EC-FOS, we then went on to validate that it was present in physiological biological fluids. So from healthy controls, we obtained some CSF and serum, and we could detect EC-FOS in these fluids as shown here. We also then went on to look at the where EC-FOS was present intracellularly. As I mentioned, it's obviously synthesized intracellularly, and we compared this with canonical FOS. And while canonical FOS is present to a significant degree, and this is wild type in each case, about 60% of cells had nuclear canonical FOS, but EC-FOS, the percentage was much less. So unlike canonical FOS, it's not present to a great degree in the nucleus, which would suggest it has different biological functions. We've also looked to see whether EC-FOS was associated with stress granules, which would be similar to canonical FOS. It was. We looked at a couple of markers, HUR and G3BP1, but it was to a lesser extent than canonical FOS, and the mutants were also not significantly more recruited in the inclusion. So with canonical FOS, the mutants are preferentially recruited into stress granules. But what we found is that when the time point when aggregates appear, that these EC-FOS is co-localizing with these stress granule markers. So it seems like the aggregates are mainly co-localizing with stress granule markers. Now, given that EC-FOS is extracellular, it would suggest it passes through the secretory organelles, so the ER and the Golgi apparatus. Because if it's a classically secreted protein. So we looked at this by immunostaining for markers of the ER and the Golgi, both calnexin and GM130, and we detected co-localization of EC-FOS. This is with the specific antibody in both compartments. So this suggests it passes through the classical secretory pathway. And N-linked glycosylation, so N-linked carbohydrates, is a common modification of extracellular proteins that are classically secreted. This hasn't been described before for canonical FOS, although O-linked glycosylation has. So what we did was use an enzyme PNGase F. So this basically cleaves N-linked oligosaccharides from proteins at the asparagine attachment site. So it would remove all a whole range of different carbohydrates, the whole spectrum. And if we treat extracellular samples, so the medium and serum, human serum with PNGase, you can see that we see this shift in size, suggesting that EC-FOS is glycosylated. We examined this further by doing glycomics, mass spec in collaboration with Nikki Parker at Macquarie, and her team were able to detect a whole range of different carbohydrate structures in the sample, so high mannose, hybrids, and complex oligosaccharides. So these structures are, again, consistent with transit through the ER and Golgi compartments. And so this confirms that EC-FOS is glycosylated, but also it transits through the classical secretory pathway. And another obvious question, of course, with an extracellular protein is, could EC-FOS be present in extracellular vesicles? Canonical FOS has been detected in exosomes, so we assumed that we would find EC-FOS also in exosomes. But what was interesting is that we looked at a range of different vesicles, which are different sizes of extracellular vesicles, representing exosomes, microvesicles, and apoptotic bodies. And whilst we could detect the presence of EC-FOS in total cell lysates, which is the first lane of each of these gels, but we couldn't detect any EC-FUS in any of the subsequent samples. So basically, EC-FUS is not present in any form of extracellular vesicle. So that was an interesting question. What's an RNA binding protein doing extracellularly, and it's not present in extracellular vesicles? Many studies of, or most studies of extracellular RNA really involved extracellular vesicles. When we look at the RNA binding properties of canonical FUS, it's fairly complex and unusual. So it's mediated by the C terminus. So it seems to be the RRM and the zinc finger domain that mediates RNA binding. But there is contribution from the RRG domains, which also increase the affinity for RNA. And this nice study from a few years ago, really characterized the binding of FUS to RNA. So basically, it seems to involve a sort of bipartite complex mechanism. So the RRM domain and the zinc finger have different RNA binding specificities. So the zinc finger recognizes this GGU motif, whereas the RRM domain recognizes this unusual RNA stem loop structure. And the RGG domains destabilize the RNA, which promotes further binding, and the RNA binds further. So given that EC-FUS contains these regions, we wondered if EC-FUS would bind RNA extracellularly. And this is an area that we're working on at the moment, but just to show you some data, we've done cross-linking and immunoprecipitation. And what we've done is basically taken some conditioned media from 5Y cells. We've performed UV cross-linking, then immunoprecipitation with our EC-FUS antibody, treated with RNAs, and then add an adapter to visualize it. And what we found was that EC-FUS appears to be binding to RNA extracellularly. So here we've got the EC-FUS band. You can see a strong signal with UV cross-linking, but that's degraded when we treat with RNAs with increasing concentrations. And what's interesting is that most extracellular RNAs, about 90%, are actually not membrane bound in extracellular vesicles, but technically it presents various challenges to work with. There are extracellular RNA binding proteins that are now being described, and there's particularly a ribonucleoprotein complex that's thought to protect extracellular RNAs. So our data suggests that perhaps this is what EC-FUS is doing extracellular, but as I said, we're doing more studies in this area. Particularly, we're trying to look at the types of species of RNAs that are bound to extracellular FUS using iClip. So with our specific antibody, we then went on to look whether EC-FUS is dysregulated in ALS. And we looked first in lumbar ALS patients and controls, and also CSF. And what we found was that in each case in ALS, we found an upregulation of EC-FUS compared to controls. And that brings me on to some work that was kindly funded by FightMND. So given we have this specific antibody and we found that EC-FUS is dysregulated in sporadic ALS, we wondered whether EC-FUS has potential as a biomarker for ALS as a whole or for different subtypes of ALS. And we obtained serum from the Macquarie Neurodegenerative Diseases Biobank. So we have a very large biobank at Macquarie with a very large range of samples, with longitudinal samples that have extensive clinical information available. And the data is not finished at the moment. It's being currently analysed by Edwin, who's our statistician. But what the preliminary evidence seems to show is that unlike in spinal cords and in CSF, in serum, EC-FOS is actually down-regulated. So this is the data we've done from longitudinal analysis. But what is interesting that we see this significant difference between males and females in terms of ALS, in terms of EC-FOS, which is sort of a confounding variable that it's taking Edwin a bit longer to analyse statistically because of that. So finally, the last thing I want to talk about is some work that we've also done using our EC-FOS antibody in patient tissues. So we wondered whether EC-FOS was associated with the pathological inclusions and also where it's localized. So our data from cells seems to suggest it's present in the secretory pathway. So we looked to see whether it was present in these compartments in patient tissues. And this work was done by Isidre Ferrer from the University of Barcelona. And what we found was that EC-FOS co-localized with a rough ER marker, REAP6, but it didn't co-localize with RAB8, which is a marker of the trans-Golgi network. And this is in the spinal cord. So this suggests, again, it's present within the ER, within the secretory pathway. This was also consistent when we looked further, and this is in the spinal cord, both ALS and control. So again, we see this granular pattern that is characteristic of the rough ER in a range of different subtypes of neurons. Interestingly, we didn't see any association of EC-FOS with pathological, the typical FOS inclusions in ALS. But what we did see was that FOS associates with the pathological inclusions in Alzheimer's disease patients. So we see strong EC-FOS immunoreactivity in neurofibrillary tangles, in dystrophic neurites around plaques, as you can see in this image. This is in the hippocampus, and we've also looked in the cortex. This suggests that our isoform is specifically associated with these Alzheimer's disease pathological inclusions. So canonical FOS has not been detected before in the Alzheimer's inclusion. So this is quite an interesting result. So to summarize what I've talked about today, we've discovered a new isoform of FOS that is extracellular, so that's unusual for the main pathological proteins in ALS, produced by an unusual alternative splicing outcome. It's incredibly misfolded, it's very aggregation prone, and the ALS mutations, the common ALS mutations, induce toxicity and further aggregation. It's present within the classical secretory pathway and it's N-glycosylated. So this is in contrast to the normal cellular FOS, which is basically cytoplasmic and mainly nuclear, but cytoplasmic in ALS. It binds to extracellular RNA, but it's absent from extracellular vesicles. It's dysregulated in ALS and it's decreased in serum. So we're examining this as a biomarker, and this is also in collaboration with Mary Louise Rogers. But EC-FOS associates with the pathological inclusions in Alzheimer's disease. So just leave you with this thought, really. So we hear a lot about proteostasis in ALS, but we talk mainly about intracellular proteostasis. So the existence of this highly aggregation-prone isoform gives the suggestion that in ALS, perhaps extracellular proteostasis should also be considered, and there is already some evidence for that from different types of pathological events. So finally, I just wanted to thank everyone involved in the work. So thanks to my team at Macquarie, and thanks to Marta, previous PhD student, and everyone else who's been involved in this work. Thanks to Yoni and Ricky, collaborators from the Quick Institute in London, Mary Louise from Flinders, Leslie from La Trobe, Isidri and colleagues from the University of Barcelona, and Ellen and Winston from the Asbestos and Dust Diseases Research Institute, and also the funding bodies. Thanks very much for listening.
Great, thank you so much, Julie. That was a really fantastic talk, and I'm really good. Interesting to hear about these sort of new developments that are coming out of your lab. So now we've got time for questions and discussion. So if anyone has anything they'd like to ask Julie, please feel free to use the reaction button to sort of raise your hand, and we'll be able to enable your microphone and camera, and you can ask your question, or if you'd like, you could also type it into the chat. Well, just while we're waiting for any questions from the floor, Julie, I'd like to kick off with a question then. I was just wondering about what you might know about the biological function of EC-FUS. What kind of RNAs does it bind, and does that give clues to what it might do if it's distinct from the canonical FUS?
Yeah, I mean, that's a really interesting question, Sarah. I mean, we're looking at that at the moment. We've just started to really examine the RNA binding properties of EC-FUS, and Cyan in my team is looking at this. We're doing some iClip studies where, I mean, that's been studied extensively for canonical FUS. So for EC-FUS, we're going to look both intracellularly and extracellularly and look at the types of RNAs that are bound. I mean, canonical FUS, seems to have a preference for binding long introns. So we have no data on that at the moment, other than we know it binds RNA extracellularly. It's possible, of course, that, being extracellular, it's involved in cell-to-cell communication. We haven't really got any evidence for what it might do, or that it is transferred from cell to cell. We've done some studies on that, but nothing to report yet at this stage. But yeah, that's a really interesting point. You would imagine it would have a different role from canonical FUS, given its distinct properties and location.
Yeah. It sounds like it's very much an unknown at the moment.
Yeah.
Right. Is there any other questions from the floor? Okay, I might ask you another question then. Sorry, hang on. My colleague Davor has a question for you.
Sure, let me turn the camera on. Thanks, Julie. It's insightful presentation there on EC-FUS. Just looking at your kind of data on biomarkers at EC-FUS and being potentially used as a biomarker for MND. Do you know if and I understand this is quite innovative stuff, but do you know if it's across other diseases like Parkinson's, where EC-FUS may be affected as well, or dementia as well, would it, just to get your view on whether EC-FUS would be a conclusive and specific biomarker for MND, or would there be some overlap as well with other disorders?
That's a really good question, Davor. I mean, that is something that we're about to look at. Actually, we've got in collaboration with Mary Louise and David Schultz from Adelaide. They've got a whole heap of neurological controls. We've only tested it so far with healthy controls. So we've got samples from other diseases, and so we'll look at its expression. It's possible it's dysregulated in other diseases as well. We have no idea at the moment, but that's something we're about to test. I mean, the fact it's present in Alzheimer's inclusions, of course, suggests that maybe it's also dysregulated in Alzheimer's. But yeah, we haven't got any results to suggest either way at this stage.
Yeah.
Thanks, Julie.
You indicated that it's obviously very prone to aggregation. Does this you see this occur intracellularly, but does it occur extracellularly as well?
Yeah, I mean, it's kind of hard to look at that. We asked Isidri did the staining in tissues. I kept asking him, can you see any evidence of extracellular aggregates? And he didn't really, but yeah, I mean, the thing is the extracellular space is even more prone to aggregate. The conditions, it's even more prone to aggregate than intracellularly. So we haven't really looked at that, but yeah, it's an interesting question for sure.
Oops. I don't think we have a bit of I think we might have a bit of a shy audience today. We don't really have any questions from the floor.
Yeah. Oh, there's one I can see in the chat.
Oh,
Do you think RNA is required for RNA EC-Fus aggregation and its interaction with other proteins such as NfO? Yeah, that's a good point. Quite potentially, I mean, that is the case, I guess, for TDP. So yeah, that's also something that we're interested in looking at. I would imagine it probably would be, given its its disordered structure. So yes, good question.
There's another question in the chat here. Are NFTs that are positive for EC-Fus extracellular?
I don't. Yeah, I'm not sure actually. I'd have to ask Isidri's opinion on that. I mean, from what he said, most of what we've seen are intracellular, but across all the samples, yeah, that's a good question. Yeah, we don't know.
Yeah. All right. Well, thanks, Julie. I think we might wrap it up there if there's no other questions. But please feel free to follow up with Julie herself, or if please get in touch with us if you need us to help you connect with Julie. Well, thank you so much for joining us today and sharing your results into this really interesting study, and look forward to seeing how it plays out in terms of the biomarker development in this space. This seminar today has been recorded, so if you do know anyone who wasn't able to attend live but would like to catch up, do let them know that this will be available on our website in a few days' time. So thank you for joining us today for the May FightMND Scientific Seminar. We hope you've enjoyed today's presentation. Please join us again next month when we will have a new speaker. Keep a lookout in your emails for news on this seminar, which will take place on Tuesday, June 23rd. Until then, bye, Beanie, and we'll see you soon.
Thanks.
Thank
Bye.
you.